Next, we'll be privileged to hear from Dr. Peter Kang. Dr. Kang is a pediatric neuromuscular neurologist and physician scientist. He's the director of the Paul and Sheila Wellstone Muscular Dystrophy Center at the University of Minnesota, where he also serves as professor and vice chair of research for the Department of Neurology. His research has focused on solving patient-related genetic and genomic puzzles in muscular dystrophy for nearly 2 decades, including the discovery and characterization of novel muscular dystrophy genes. More recently, he has been working on a collaborative project to translate cell therapies for muscular dystrophy into human clinical trials. So, Dr. Kang, we welcome you up here.
So I want to thank the MDA for sponsoring and organizing this conference, and then also our industry partners who were mentioned before. We really appreciate their presence. They're really helping to lead a lot of innovation in this field. So I wasn't sure who would be in the audience and what disorders people would most want to hear about. So I'm going to try to cover a bit of a spectrum as much as possible, and then also go into a depth in a few topics. So these are my disclosures, and to be fair and balanced, I'm going to avoid naming any company names or brand names during my presentation. So we'll talk a little bit about shorter diagnostic journeys, classification, FDA-approved therapies, which has been a very active area, and then therapies in development, which is also a very active area. So shorter diagnostic journeys, this is all what we'd like to achieve because the sooner you can get to diagnosis, the sooner you can look at therapeutic options. So Dr. Karachunsky discussed newborn screening in depth, and that was a fabulous presentation. And I feel that genetically based newborn screening is the wave of the future, and this will shorten many diagnostic journeys, although it'll raise new challenges, and I have an example of that I'll show. For now, we still diagnose many neuromuscular disorders when children and adults become symptomatic. So this is the famous diagram of Duchenne's first description of Duchenne muscular dystrophy from the mid-19th century. And if you look at a photo of a more recent patient, you can see how accurate that diagram was, and you can see a lot of the same features. And then the Gower sign, which is another very well-known physical finding, was described in the late 19th century. Duchenne's a little bit less common than we thought it was previously. If you look in the literature from the 20th century, the figure that you see a lot is 1 in 3,500 boys who are born. It's probably a little closer to 1 in 5,000 based on more recent epidemiologic data. And I think a lot of you know that the onset is typically during early childhood with symptoms of gait difficulties, frequent falling, and toe walking. There's often no family history. So how is Duchenne diagnosed? One of the biggest advances occurred actually in the mid-20th century. Before then, it was really just describing the physical features, and that's all we had to go on. But in the mid-20th century, investigators noticed that this enzyme called creatine phosphokinase was elevated in patients with muscular dystrophy. Not only that, you could sort of distinguish between different types of muscular dystrophy based on how highly it was elevated. So that really helped, and it's something that we do all the time in clinic even today, but it was around this time when it was developed. You can sort of get clues about different types of muscular dystrophy based on the CK level. It's not very precise, and there's a lot of overlap. And this is just a table from a textbook chapter from a few years ago illustrating the ranges that you see in various types of limb-girdle muscular dystrophy. Another issue that comes up is now that we know that certain enzymes are elevated in muscular dystrophy, sometimes we stumble across patients accidentally. In a lot of cases, maybe not a lot of cases, but in a number of cases, people get liver function tests checked either because they're starting a new medication or for some other reason that's fairly routine, and all of a sudden they're elevated. And then some of these patients actually have muscular dystrophy, but then unfortunately they go on a prolonged diagnostic odyssey. Sometimes they even get a liver biopsy before they're diagnosed. A little over a decade ago, we tried to figure out what the expected ratios were for some of these enzymes, the CK and the ALT and the AST. This is a fellow that worked on this, Hugh McMillan. He's now a faculty member at the University of Ottawa. And he led this study and did a really amazing job. And this is a formula that I've used in clinic myself, and that's very helpful to see, is everything okay with a child's liver who has muscular dystrophy? And then there's muscle biopsy, which some of you in the audience may have gone through, and this is becoming a little bit less common as a diagnostic test. It's really good for evaluating myopathies. It can also detect some nerve problems. So back before we knew about specific genes and proteins, these were some of the basic stains that were available. And so on the left is normal muscle biopsy. On the right is one from a patient with Duchenne muscular dystrophy. And you can see the dramatic difference, but we couldn't pinpoint specific diagnoses just based on this alone. So through a combination of various techniques, up until the late 20th century, we were able to diagnose a lot of patients with fairly good accuracy, but not precisely. So all of this changed in the 1980s when Lou Kunkel's laboratory found the gene for Duchenne muscular dystrophy. This was really a landmark, and it opened up a whole wave of genetic discoveries. And now there are dozens and dozens of genes known for muscular dystrophy, but this was the first. And so because of that, and the discovery of the protein the year after, now we're able to look at dystrophin staining and actually pinpoint the diagnosis. Even if we don't know the mutation, we can tell by the biopsy what's going on there. And this was replicated for other genes. This is a paper we published almost 20 years ago with a series of patients with gamma sarcoglycanopathy, which is one of the limb-girdle dystrophies. So genetic testing has just exploded. The discovery of the human, maybe not the discovery, the sequencing of the human genome in the around 2001 really opened up a lot of doors in terms of diagnostics. And so our genetic testing modalities have advanced very rapidly. Previously, we had karyotypes, southern blots, PCRs, and those were all very helpful, Sanger sequencing. And now we have chromosomal microarray, next-generation sequencing, and in my laboratory, we're exploring third-generation sequencing, which is becoming increasingly adopted in clinical test laboratories. No genetic test method is perfect, so we're still using combinations of these approaches. One thing to keep in mind for anybody who's registered for a research study, and we do research on undiagnosed patients in my laboratory, so you're welcome to reach out if that's of interest to you. There is a difference between clinical genetic testing and research genetic testing. In clinical genetic testing, there's an organization called CLIA, and that's a certification body, and it certifies that the diagnostic test laboratory is following good practices and that they're doing things accurately and that they're being reliable. And so it's important to issue genetic test reports. You need the CLIA certification. So from a CLIA certified lab, you get an official report. It goes in your medical record. Research testing is a little bit different. They can use different techniques. We use a lot of nanopore sequencing, which is third-generation sequencing in my laboratory. We're not CLIA certified, which means that we're not allowed to issue official reports. We are able to relay information to clinicians who are taking care of patients so that they can get confirmatory testing done, and we've done that on a number of occasions, and that's helped some patients get to their diagnoses. So nanopore sequencing is something that we really use a lot in my laboratory, and it's pretty exciting because we can look at DNA strands and sequence them without having to create extra copies of the DNA and potentially introducing distortions. And so these strands of DNA are run through these little nanopores that are on a chip, and then the pores read the electrical currents that change as the DNA sequences go through, and then from the electrical current patterns, you can figure out what the DNA sequence is. It's just amazing what this is capable of doing. We did a pilot study that we published just last year. We looked at 10 families with muscular dystrophy that had various diagnostic puzzles. Some of them had no diagnosis. Some of them had a suspected diagnosis, but it was incomplete. And so we looked at them, and we were able to find some helpful information on several of these individuals. So I'll skip ahead a little bit. So this was a patient who had Duchenne muscular dystrophy, but no mutation that was identifiable. And it was clear that this is the correct diagnosis because he didn't have any dystrophin on his muscle biopsy. And he had clinical testing, and nothing was found on it. And what we discovered when we did the nanopore sequencing was the reason why no one could find this is that a big chunk of his DNA in the dystrophin region was actually flipped, and that's called an inversion in genetics. And so if you just sequence little sections of DNA, it looks totally normal. But if you look at the whole thing, or a big stretch, which is what the nanopore sequencing does, you actually see what happened. And so we saw that, and then the bottom images there just show you the confirmatory testing. We did some PCR analysis around the breakpoints to just make sure we were seeing what we thought we were seeing. Another one of these 10 patients was an asymptomatic individual who looked like he might have had Duchenne muscular dystrophy. However, he was an adult male, and he worked in a physically intense occupation and had a normal CK level, yet he had a duplication in his dystrophin gene, which was very odd. And so we were wondering, is the duplicated region of dystrophin somewhere else, and was it accidentally put in another chromosome early in development? Or was it really part of the original dystrophin gene? Because that would help us with interpreting what was going on here. So we found that, in fact, it was right there in the dystrophin gene. Both of these duplicated segments are right next to each other. And so the interesting thing that this raises, the interesting question this raises is, if we do start doing genetically based newborn screening in the future, which I suspect we will, we'll encounter cases like this, and we have to be careful about how we counsel patients and interpret the results and give patients prognoses. So it's very worthwhile to pursue this and to identify these types of cases, because that'll help us anticipate some of these dilemmas as we expand and deepen newborn screening processes. And then the last case that I'll show from that publication is a congenital muscular dystrophy case. So LAMA2 is a gene that's found in the recessive state when it's mutated, meaning that both copies of the gene have to have a mutation to have the disease. What happens in some patients is that you only find one mutation. This patient looked very much like they had the congenital muscular dystrophy, and the clinical testing couldn't find the second mutation. So we did the nanopore sequencing, and we found the second mutation. It was a duplication of exon 30, and that helped solve what was going on with this patient and kind of solve that little mystery. And this is a similar pattern of evaluation that we did. We just used PCR to make sure that the duplication was where we thought it was. So this is something that we continue doing in my laboratory, and we're expanding the use of this. It's a technology that I suspect will be increasingly used in clinical settings. It is really powerful, and it can apply to a broad range of different mutation types. So let's talk about classification for a minute, or a couple of minutes. So there are, in the neurology world, we try to divide up disorders anatomically. So for the neuromuscular neurologists, we try to divide things up by whether it's in the motor neuron disease category, neuropathy, neuromuscular junction, or muscle disease. We do have, in addition to our faculty at the University of Minnesota, our physical therapists and nurses and many talented people who are here and will be sharing their knowledge, we have some great experts who've joined us, Dr. Escuri from Gillette Children's Hospital, who will be presenting a little bit later, and then Dr. Michael Shai from the University of Iowa. So he, as some of you know, is extremely well known in the field of Charcot-Marie tooth disease. So it'll be very exciting to see all of these future presentations in the rest of the day. But this kind of just gives you an idea of how we think about how these disorders are categorized and classified. This is a diagram of part of a muscle fiber, and the top is the outside of the muscle fiber, and the bottom is more the inside. And these are all proteins that are linked to different muscle diseases, mostly muscular dystrophy. And you can see that there's been an explosion in knowledge. We know so much more about how the muscle functions and how dysfunction of some of these proteins can lead to disease. It's helping us to understand both how the muscle should be functioning and how things can go wrong when one of these proteins is not doing what it should be. For classification, these are the standard classifications of muscular dystrophies, and some of you may be interested in particular ones of these. And they vary in severity and also frequency. There's been a lot of classification confusion and issues with limb-girdle muscular dystrophy in particular, and this is what I study. There are over 30 associated genes for limb-girdle dystrophy, so this is something that has been kind of a dilemma. When the first limb-girdle genes were described in the 1990s, a classification came up where you divided them into type 1 for dominant and type 2 for recessive, and then you add a letter at the end. So the first recessive one was just called type 2A, second one 2B, and we were doing pretty well for a couple of decades. The problem is we ended up at limb-girdle 2Z and ran out of letters in the alphabet, and so now people were kind of puzzling over, now what do we do? Do we call the next one limb-girdle 2AA? And so that became apparent that this is not viable. So there was a workshop a few years ago where there was extensive discussion, and a new classification system has been proposed, and it's been widely adopted. There's limb-girdle 1 that's now limb-girdle dominant, and then limb-girdle 2 became limb-girdle recessive, and now there are numbers at the end, and so we don't have to worry about running out of numbers. So there have been some switches in terms of what's assigned to what, but in general, a lot of the same order has been followed. A lot of people, and I've noticed patients in particular, when they were diagnosed originally with one name, like limb-girdle 2B, it has become part of their identity, so I think it's sometimes a little tough to switch to thinking about it with a new term. So a lot of people are using both terms just to make everybody comfortable, and so I think we'll probably keep doing that for at least a little while. And this is just some of the common limb-girdle muscular dystrophy genes. These are common and classic congenital muscular dystrophy genes for anybody in the audience who has an interest in this. And there are 3 broad categories of these. And there's increasingly some recognition that the dystroglycanopathies and the collagenopathies, actually all of these, have some overlap with the limb-girdle dystrophies. And then the last set of genes, just to try to be a little bit more broad, these are other genes associated with muscular dystrophy. And so our knowledge base has really exploded in terms of what we understand about this. Of course, there's a lot more that we want to discover. So FDA-approved therapies. Now this, when I was mentioning to someone earlier this morning, when I started my career, this would have been a blank slide, and I would have had to move on to animal experiments, which is what my presentations were like in these types of settings back then. What's really nice to see over the 2 decades of my career is that there's been an explosion of FDA-approved therapies for a whole variety of disorders. So spinal muscular atrophy, I won't dwell on too much because Dr. Karachunsky's spoken about this already. There are 3 approved therapies, all of which have pluses and minuses. So for every individual patient, a different decision will be made. And so it's nice that there are multiple options. And this is from a review that a neuromuscular fellow and I published just last year, just showing some of the features of the 3 therapies that are approved. Pompe disease, this is one where there has been an approved therapy for a while, L-glucosidase alpha, and that's an enzyme replacement therapy. So Pompe disease is an enzyme deficiency disorder, and it has limited uptake in skeletal muscles, and so you need to administer high doses of this. The other thing about enzyme replacement therapy is that you'd have to get IV infusions on a regular basis for your entire life once you start this. So it is something that you have to think carefully about what the pluses and minuses of this are. There is a new enzyme replacement therapy that was just approved a couple of years ago, and it's a slightly different molecule, and it has enhanced binding to a specific receptor that's really important for therapeutic efficacy, and so that's improved clearance of glycogen. So it's not quite as broad as the original enzyme in terms of what settings it can be used for, but it's something that is a nice addition to our toolbox. There is one FDA-approved therapy for periodic paralysis, dichlorphenamide, for anyone who has an interest in this. It's a carbonic anhydrase inhibitor. It's an oral medication. There are other medications that have been used for this disorder for a number of years, but those are off-label because they're older medications. So it's certainly worth considering the older medications, but this one is the one that's gone through the FDA approval process. And just this year, there's a first FDA-approved therapy for Friedreich ataxia. and this is an oral medication also, and it's for those ages 16 to 40. Dr. Karachunsky mentioned that we have 6 FDA-approved therapies for Duchenne muscular dystrophy, so we've got quite a variety, and the most recent of which was this year when delandistrogene was approved, which is a gene therapy. There's been extensive studies of antisense oligonucleotide therapy. Out of the 6 therapies approved for Duchenne dystrophy, 4 of them are exon skipping or antisense oligonucleotide therapies, and these are short strands of nucleotides that bind to pre-mRNA and alter the splicing patterns. So these help change the mutations that are out of frame into ones that are in frame, and that helps alleviate the severity of these disorders. Gene therapies had received a lot of attention recently. So just to give you sort of the spectrum of what's been approved for human use, and this is using the AAV vector strategy. The first was for Leber congenital amaurosis. The next was spinal muscular atrophy, which is one of the neuromuscular disorders. There are 2 more for hemophilia, and then the Duchenne muscular dystrophy therapy that was just approved this year. So let's talk about therapies in development. There's a lot more gene therapy that's coming down the pipeline. There are multiple other therapies for Duchenne muscular dystrophy that are undergoing human clinical trials. There are multiple therapies for limb-girdle muscular dystrophy. These are all gene therapy strategies that are undergoing human clinical trials. And then there's one for FKRP deficiency, another one of the limb-girdle dystrophies, and then for Pompe disease also. And there may be others that are not on this list. There's been an incredible proliferation of these studies. So let's talk a little bit about this. I think a lot of you have heard of AAV and gene therapy. So where does this all come from? Basically, AAV is a package. It's basically a little vehicle for delivering pieces of DNA. There are lots of delivery vehicles that have been used and are studied, and you can see this list here. Previously, adenovirus was tested as well as lentivirus. So adenovirus triggered a very bad reaction. This is going back about a quarter century. And it's used on a very limited basis in certain cases, but for standard gene therapy applications, it's not a good choice. Lentivirus, one of the issues with lentivirus is that it integrates into the genome and can cause some unwanted effects. It's used in specific circumstances when you're doing combined cell gene therapy, but not for just delivering straight virus into a human. So that leaves us with adeno-associated virus, which I expect is going to be the vector of choice for quite some time for many applications. So AAV is not exactly adenovirus. The 2 are very different, and they're actually not that related biologically. It so happened that AAV was found when people are trying to isolate adenovirus. They found these little viruses next to them and were wondering what they were, and then finally realized that they were a different species of virus. The way AAV is transformed and the way it's made, it's not able to replicate by itself, and that's really important for safety reasons. You know, it'd be nice if it could replicate in the body and propagate the effect, but at the same time, you increase the risk of unwanted effects if it replicates. So one of the nice things about AAV is that it doesn't replicate, doesn't integrate into the genome. And so you take some of the genetic material that was in the wild-type AAV, meaning the naturally occurring AAV, and you replace it with a promoter and the transgene that you want to deliver, whether it's SMN for spinal muscular atrophy or microdystrophin for Duchenne muscular dystrophy, or one of the genes for limb-girdle dystrophy or Pompe disease. So that area where the transgene is where basically the cargo goes. Developing these AAVs is not trivial, and one of my colleagues, Christina Pasak, is an expert in this, and spends a lot of time focusing on things like adapting the capsid, meaning the whole package of the AAV, the design of the vector sequence, the dose, the delivery route. Those are all issues that have to be addressed before you even think about putting them into humans. So it's just amazing that there are so many human clinical trials going on, because that means that all of that work was done over the past decade plus to optimize these packages. Small molecules are important to remember. Those are small molecules are basically a fancy name for traditional drugs. You know, it's like aspirin or Tylenol. Those are small molecules. Those can also play a role in the future. It's not only the biologics. So for Duchenne and Becker, there are a couple of treatments that are undergoing human trials and are investigational. For limb-girdle muscular dystrophy, there are ones that are being investigated, and these are different types of molecules that act in different ways, usually in the molecular pathway that's a key part of the disease. One nice thing about small molecules is that a lot of them can be just taken by mouth, so that's something that's very convenient for patients. So even if you have to take it every day, it's something that's relatively easy to take. Nitric oxide agonists were explored more about a decade ago. I think there's a little bit less going on right now on it, but they were shown in some mouse models to help with muscular dystrophy. Myostatin inhibition has been pursued for the past 2 decades because it's a gene that slows down muscle growth. And so the thinking is that if you inhibit this gene, that you'll augment muscle growth, and that could apply to a wide range of muscle diseases. So there have been trials that have gone on. They have not shown good efficacy yet, but this is an approach that's still being studied. And then CRISPR-Cas9 is incredibly exciting, and this has been in the news a lot also. You can basically go in and edit genes and change them back to where you think they should be. And so in cells and in animal models, this has been a really powerful approach. There has been one study in a human clinical trial. Unfortunately, it led to a fatality in 2022, so I think we have to take a step back and kind of rethink this before we try again. I'm sure that there will be future clinical trials, but it'll require a little bit of recalibration. So I would like to talk about stem cell therapy for a little bit. This has also had a long course. For muscular dystrophy in particular, there are human trials back in the early 1990s. Unfortunately, they had disappointing results, so then the activity around this quieted down for a little while. But there have been ongoing experiments in mouse and dog models that really suggest that there's significant therapeutic potential. The nice thing about stem cell therapy is depending on what type you use, you might be able to apply it to a broad range of disorders. So the University of Minnesota does have a really strong program in stem cell biology. And so we've been making an effort for quite a while, and it's really starting to get closer and closer to human clinical trials. So this is a group that's put together in collaboration both with the Muscular Dystrophy Center here at the University of Minnesota and then other entities within the universities. So developing a cell therapy is very complex. Rita Perlingaro has been really been leading this preclinical effort and has worked on this for over a decade. And we're getting closer and closer to a human trial. Just to kind of describe, I think some of you may have heard about some of these transplantation strategies, and so there are stem cell treatments that are being used on a daily basis for bone marrow transplants for certain disorders, but they've not been optimized for muscular dystrophy or other neuromuscular disorders. So there are 2 basic ways you can approach a cell therapy. One is called allogeneic, and that's on the left, and then the other is autologous, which is shown on the right. So I'll go from left to right. So allogeneic means that you're getting the transplantation from a donor that's usually not related to you. It may be, but it's typically unrelated. And so you take a healthy donor, and for this approach, which is iPSC, induced pluripotent stem cell development, you take the healthy donor, you take some cells out, and you reprogram them into those iPSCs. The thing that's amazing about iPSCs is that they are pluripotent stem cells, meaning they can differentiate into a whole range of different cell types. And so then they differentiate, and Dr. Perlingaro has been able to transform these into healthy myogenic progenitors, meaning they're primitive muscle cells, and then they're transplanted into muscular dystrophy patients. So that's one approach, and that's the one that we're focusing on right now. The other approach is the autologous transplantation route, and this has really been used to great effect in some other disorders. You take cells out of a patient, their own cells, and you reprogram them, and then you actually correct the genetic issue in those cells, and then you transplant them back into the patient. So So that's a slightly different approach. The end effect in both cases is that the patient's getting healthy cells. So we're using the allogeneic approach. There are pluses and minuses, as you can imagine, of both approaches. We like the allogeneic approach because it is potentially applicable to a larger number of people more quickly, meaning that a single donor could potentially treat a lot of patients. Both of these approaches have their utility in various settings. So it just so happens that right now we're focusing on allogeneic. So this is the roadmap that we've been following for clinical translation. And all of this preclinical work has been done either by Dr. Perangillo's lab or collaborators here at the University of Minnesota and collaborators at other institutions. So we need a methodology to generate the cell product because if we want this to be approved by the FDA, we have to have a reliable way of generating this. This shouldn't be a one-off. It should be a standardized procedure. We also need to purify the cell product and then generate GMP compliant product. GMP is good manufacturing practices, and so we really want it to be clean, safe for humans to be able to receive these. And then we want to characterize them to make sure that they are what we think they are, and then transfer them to a clinical grade facility. And then we do use lentiviral vectors. I did mention that lentiviral vectors are used in certain settings a few slides ago, and so this is one of those settings. And then production grade, clinical grade cell product, and then GLP preclinical studies. And then the last step, which is what we're going through right now, is an IND filing with the FDA. And we hope that we will be able to get that approval so that we can start a clinical trial in the near future. So just to kind of show this graphically, I went over this in the previous slide, but we're basically at that very last step before we can start a first-in-human clinical trial. So induced pluripotent stem cell derivatives have been used in humans in the past, but not specifically for muscular dystrophies. That's why we're calling it first-in-human. So we have thoughts about a phase 1 and then a phase 2 human clinical trial. We are starting off with individual muscle injections. One of the issues that's come up with cell therapy is that it's difficult to administer it systemically to spread through the whole body for a variety of technical reasons, which in the future we hope to overcome. But for now, we're doing individual muscle injections. So we want to look at feasibility and safety of these types of injections in patients with Duchenne muscular dystrophy, and then we want to, in our phase 1 study, recruit a number of individuals to participate. And phase 1 will involve an injection into a foot muscle. We're thinking tentatively that in phase 2 we might start looking at hand muscles. Once the initial safety is established, we want to think about, is this applicable to other muscular dystrophies? And this therapy is not necessarily specific to Duchenne, so it could include limb-girdle, FSHD, and a range of other neuromuscular disorders, and look at additional muscle injection sites. And then we will kind of think about in the future, is there any way to make this a little bit more systemic so that we can treat more muscles with a single injection? And then one last topic I want to cover is a network that we participate in. It's called GRASP-LGMD, and I just want to show this as an illustration of how important it is to have friends and to work with other centers around the country and in other countries, because a lot of these diseases, in fact, I think all of them are rare diseases, meaning that it's very hard to collect enough patients in a single center to conduct a major clinical trial. So it's really important to work with other centers. So the University of Minnesota is part of this GRASP network that focuses on limb-girdle muscular dystrophy. It's led by Virginia Commonwealth University, Nick Johnson being the overall PI, and it has funding from a variety of sources. So this is just a map that shows some of the centers around the country and a couple in Europe. What this consortium is doing is gathering natural history data, optimizing outcome measures, and providing a platform for multi-center clinical trials. And we have several studies ongoing, as you can see. So I want to end by just showing some of the team members who are working towards developing new therapies and improving diagnostics. So this is the leadership team for the MD Center, and you just heard from Dr. Karachunsky. Dr. Avasti is our research director, and he's been studying muscular dystrophy for quite a long time and is well known throughout the world. And we're really glad that he's part of our leadership team. We've got an amazing team, some of whom are here today, that help with various aspects of our research and our clinic operations. And so this was a photo that was taken just earlier this year. This includes both clinical team members as well as research team members. And so it's really nice to have that collaboration and teamwork amongst all of these individuals. And so these are some of the people who've been working at our MD Center and MDA clinic. This is just a photo of my laboratory taken recently, and that some of the genetic data that I showed a little bit earlier in the presentation was generated by my laboratory. And then for research support, I'm really grateful for all the support that we've had over the years. Thank you very much. I'm happy to take any questions.